Translation, that is in gene expression, how the proteins are synthesized. Yes, children. So, what are proteins first of all? So, from your class 6 or from your lower class itself, you are studying about the macromolecules like carbohydrates, proteins, fats, vitamins, and minerals, isn't it? So, now coming to proteins. So, what are the major functions of proteins? Proteins are very essential for the growth, they are very essential for the enzymatic reactions, metabolic reactions, and antibody production. Yes, and basic thing which is very very essential is body growth and development. Okay, so these are the functions of the proteins. This we learn from our primary level itself, I think so. So now how this protein is being synthesized in the living organisms, how the genetic information is carried for the protein synthesis in the cells, living cells. Okay, so let me explain that with this simulation. Okay, so this gene expression simulation, let me explain that. Okay, so now if you look at this, there are the tools, biomolecule tools have been given there, and then there are transcription factors are there, RNA polymerase enzymes have been given, ribosome is given, messenger RNA is also given, and then messenger RNA destroyer, and then negative transcription factor, everything. And there are three blocks of different shapes they have given, that is diamond shape and then hexagonal shape and the cylindrical shape of proteins. So first we will start with the gene expression for the protein synthesis. How this gene expression comes? From where it comes from? Okay, it starts from the DNA. What is DNA? It is nothing but the deoxyribonucleic acid. So in fact, it is present in the nucleus. So nucleus is the brain of the cell. This all things we know from the lower class itself. So now the cell will produce the DNA. That is the basic material for the protein synthesis. Okay. So now from this deoxyribonucleic acid DNA, the DNA will transcribe its information or passes the information to the RNA, that is ribonucleic acid. So we all know that what is deoxyribonucleic acid and what is ribonucleic acid and what is the difference between them. What is the difference between them? Deoxy means the nucleic acid which lack the oxygen in its second position. That is known as the deoxyribonucleic acid, whereas ribonucleic acid will have the oxygen present in the second position of the pentose sugar. So this all this we know about the basic structure of nucleic acids. If you people are the biology student, you are very well known about the biomolecules and their structure, I think so. So now from the DNA, this RNA will be transcribed. That process is known as transcription. So let me explain first the transcription process. So that's the first step before the protein synthesis takes place, because the RNA only is going to carry the information from the DNA and it will going to take it out of the nucleus, and in the cytoplasm only the protein synthesis will take place using the ribosomes. Okay, so now let me explain the transcription. So this formation of mRNA, messenger RNA, which is going to carry the message from the DNA for the protein synthesis. So now I am going to take the first one, positive transcription factor, and then I need the enzyme for this. For any metabolic reactions, the living organisms or the living cells need the enzyme. So here for the mRNA synthesis, okay, there is an enzyme known as a RNA polymerase. Polymerase means it will produce many, that is known as the mRNA polymerase. So now I am going to take the enzyme also. Now what happens? See, there is the mRNA synthesis taking place, and now this mRNA is ready for the protein synthesis. So now the ribosomes has to come and play the role, and here the tRNA will bring the amino acids for the protein formation. So this is the protein which has been formed here. So this is the general outline I told you. I'll repeat this again. So now I'm going to show you the next gene expression. So look at this. Again, I'm going to take the positive transcription factor. So bring it here, and we need the RNA polymerase enzyme, and before that we need the regulatory substance factor also, and then we need the RNA polymerase. Yes, so all this together will produce the, see, the DNA is converted into mRNA, that is transcription. So this is the transcription process. See, this coiled black color coil, that is known as the mRNA. Now this mRNA needs the ribosome for the protein synthesis. So this yellow color ribosome will bring the tRNA along with the transfer RNA, which brings the amino acid necessary for the codons present in the mRNA, so that the information is passed. So the amino acids, they form the polypeptide, that is first peptide bond will be formed between the amino acids, and then this peptide bond will form many peptide bonds, that is known as the polypeptide, and these polypeptides will form the peptones, and the peptones will form the protein. So this is the protein molecule, second protein formed. Again, if you have any doubt, again third gene expression I am going to show you. So again, what is the first step? This is the factors which are necessary for that, and then we need this, all the transcription factors, regulatory region, and then we need the RNA polymerase enzyme. Now the DNA is transcribed into mRNA, and after the transcription is over, the mRNA will be detached and the enzyme also detached. Now the mRNA needs the ribosomes for their translation. So the mRNA, the ribosome, and the tRNA, all the three together will translate the transcription process which has been taken place. No, that mRNA will form the protein. This protein, it is nothing but the polypeptide. So this is about the protein synthesis, which is the basic thing I have explained you in a very simple manner, children. Hope you understood about the protein synthesis. Yes. Now again one more time. So again I am going to bring the positive transcription factor, and then we need the mRNA polymerase enzyme. Enzymes are nothing but the biocatalyst. They will enhance the reaction. Now here what is the product? mRNA is formed, and the mRNA is detached from the RNA polymerase also. Now this mRNA is waiting for the ribosome. Now this mRNA came out of the nucleus. Yes, that's why it is floating in the cytoplasm. Now it needs the ribosome. So the ribosome, when it comes, it brings the tRNA, transfer RNA also along with that. This tRNA will carry the amino acid which is necessary for the translation of the mRNA codons. So now this what is that? This diamond shaped brown color is the protein. That protein is formed. Understood? So this is the protein synthesis mechanism, children. So initially the mRNA brings the information from the DNA, and from the mRNA only your protein is synthesized using the ribosomes and the tRNA. So this tRNA will bring the amino acids, isn't it? So these amino acids will form the anticodons for the codons present in the mRNA. What are codons? Codons are nothing but there is 3 amino acids will be there, and to that three amino acids, these anticodons will be paired, so that you will get a peptide. So this peptide, again they will multiply, they will form the polypeptide, and then peptones will be formed, and finally your protein macromolecule will be formed, children. This is how protein synthesis will take place in living organisms. Okay, and this protein play a major role in the structural formation. I told you body structure and development is very essential in the living organism, and it is an enzymatic role. Since this RNA polymerase, I said no, that enzyme is also a protein only. And not only that, these proteins are very essential for the antibody production and defense mechanism. Whenever we fall ill, these antibodies are produced naturally. They are nothing but the proteins only. They are also synthesized by this translation method only. Okay, so these are the functions of the proteins. Understood, children? Hope you would have understood my class very well. We'll see you in the next class, children. I hope all the concepts are very clear for you. Share and subscribe for more videos. Thank you.